A very good morning to all. I, Deepthi Gulati, working as an associate professor in the Department of Biotechnology, am very pleased to introduce you all to my ex-student and alumni of Dolphin Institute, Dr. Smijan Soman, for today's lecture. Smijan completed his master's in 2008 from Dolphin Institute and PhD in neurobiology from Cochin University of Science and Technology in 2013. He was postdoctoral research associate from August 2018 to March 2022 in the Department of Pharmacology at Reno School of Medicine, University of Nevada, United States of America, where he is currently employed as assistant professor research, a neurobiologist with more than 10 years experience in using advanced molecular, cellular, and biological approaches to pursue the discovery of central nervous system disease mechanisms. Dr. Smijan Soman is an author of more than 15 publications. In this lecture, he will be talking about the molecular mechanisms driving Parkinson's disease. Today is also a good opportunity for all of you to take career guidance from him as well. With no further delay, I call upon Dr. Smijan Soman for the talk. Thank you all.
Thank you, ma'am. Thank you for the kind introduction. Hello, everybody. I couldn't see all of you, but I hope you all are doing good. I'm going to talk today about understanding the molecular mechanisms driving Parkinson's disease using PINK1 kinase. So Parkinson's disease is the second most common neurodegenerative disease after Alzheimer's disease. It is estimated that in India, we have around 5 lakh patients, which is a very conservative census. Apart from that, if you look in the worldwide numbers, there are around 6 million people affected by Parkinson's disease. So the main symptoms of Parkinson's disease are tremor, rigidity, bradykinesia, postural disturbances. So these are mostly associated with mostly motor symptoms. So Parkinson's disease comes under movement disorder. But what people don't see and what patients suffer through is a battery of non-motor symptoms such as dementia, depression, anxiety, sleep difficulties, swallowing difficulties. They have problems with their esophagus, constipation, and there are a myriad of different symptoms which the PD patients suffer through. And biological research has to reach and look into all those symptoms as well, apart from the motor symptoms. So what is the Parkinson's disease pathology? Unfortunately, you cannot see the whole slide here because there were animations. So I'm just trying to work with what I have right now. So Parkinson's disease is caused by loss of dopaminergic neurons in the substantia nigra pars compacta of midbrain. So this is a region which, if you look, it is straight bilateral over here. So it goes into the midbrain, and it is on the both on the right and the left side. So this particular region, which is associated in the midbrain, it is mainly rich in dopaminergic neurons. So dopaminergic neurons are neurons which synthesize dopamine. So dopamine is a neurotransmitter, and it is mainly involved in a series of functions, of neuronal functions. But the Parkinson's disease patients are mainly affected by the motor symptoms, where there is not enough dopamine, which as I mentioned, is a neurotransmitter and is required for neurotransmission, is absent, and thereby it causes an alteration of neuronal function. And this alteration in neuronal function and loss of neurotransmission, it causes the various symptoms which I had described before. So what are the challenges with current Parkinson's disease therapy? So there is no cure for Parkinson's disease today. So that I want to make that clear. So the Parkinson's disease research is trying to find out various molecules, biological molecules, which can alter not only the symptoms but slow down the progression. So the present treatments against Parkinson's disease is against the symptoms. So it is symptomatic. That's how we call it. It's a symptomatic treatment. So it only tries to address the symptoms such as I previously described, motor symptoms such as bradykinesia, tremor, and all. So L-DOPA is the present choice of treatment. So you may think like, why can't we just why can't we just intravenously inject these patients with dopamine, and that may just cure the effect. Unfortunately, dopamine cannot cross the blood-brain barrier. So that's why a precursor to dopamine, L-DOPA, is given to the patients. L-DOPA can cross the blood-brain barrier, and it is metabolized into dopamine in neurons in the brain itself. So what are the shortcomings of the current treatment approach? So mainly, it does not improve the disabling motor and non-motor Parkinsonian symptoms. That is the main problem. And it is also associated with severe side effects. And one of the other major problem is it does not halt disease progression. So as I told you before, PINK1 is a progressive neurodegenerative disease. So gradually, as the PD patient ages, the disease becomes more chronic, more neurons die throughout time, and symptoms becomes worse. So what causes Parkinson's disease? We don't know for certain, that's for sure. So 70 to 80% of PD patients have something what is called a sporadic PD. By sporadic PD or Parkinson's disease, it means that we certainly don't know the exact reason why these patients are suffering from Parkinson's disease. There may be environmental factors which are contributing towards that, such as toxins, such as rotenone. So rotenone is a toxin which is found in pesticides, and people didn't know that it causes Parkinson's disease, and it was used uh, as a pesticide very commonly. And apart from that, aging, of course, aging is a factor. It contributes towards developing neurodegenerative diseases. Apart from that, there are genetic risk factors also involved. Certain mutations, certain single nucleotide polymorphisms in certain genes, which actually makes these people uh, prone to having Parkinson's disease. Apart from that, around 20% of PD patients have some sort of mutation, some sort of a gene mutation. And one of the major denominator which links both sporadic PD and familial PD is that majority of these patients and majority of these cases have some kind of a mitochondrial pathology involved. So, as you all know, mitochondria is the powerhouse of cell, but there are also many other uh, many other roles of mitochondria for the for the proper functioning of neurons, for the proper functioning of cells. So mitochondria is involved in energy production. It is involved in calcium homeostasis. It controls the levels of calcium inside the cell. There are a wide array of functions attributed to mitochondria. So one of the mutation is found in PINK1. So PINK1 is one of the protein which have focused on throughout my career. So loss of function mutation in PINK1 causes early onset Parkinson's disease. So this is early onset. So these people are young, they are relatively young, around 40 years old, and they start showing symptoms of Parkinson's disease. So this is an early onset kind of a Parkinson's disease, and it progresses, and it is because it comes very early. The symptoms are also very bad when they progress into a later age. So PINK1, what is PINK1? PINK1 is or P10-induced putative kinase. Is a serine threonine kinase. You must have studied in your biochemistry textbook. What is a function of a kinase is to catalyze. It's an enzyme, right? So it adds phosphate groups to substrates, which modify the behavior of their substrates. So this phosphorylation is ATP dependent, of course, and PINK1 is also a protein or an enzyme which phosphorylates various proteins involved in mitochondrial function, and I'll be talking about extra mitochondrial function as well. So as you can see on the right, this is like the kinase domain of a PINK1 protein, and this is a ubiquitin protein. This is another protein which is phosphorylated by PINK1. So the ubiquitin protein has a phosphate acceptor site, and when the PINK1 kinase transfers a phosphate group, a phosphate moiety, to the phosphate acceptor site on the ubiquitin, it modifies the function of ubiquitin. So there is a conformational change in ubiquitin that changes the function as well. So phosphorylation uh, is a is very important for the proper functioning of the cell, and many different cellular pathways are activated by phosphorylation, deactivated by phosphorylation. So it is very important for neuronal function. So I had previously told you about PINK1. So I'm just going to explain here what is the role of PINK1. So PINK1 is a protein which is mainly involved in what we call mitophagy. So it is the autophagy of mitochondria. So mitochondria is a dynamic organelle, and there is a lot of things going around. You know that there is energy production going on through the oxidative phosphorylation and the electron transport chain. And this is a site of a lot of free radical production. You may have heard about oxidative stress due to the free radical production. And this oxidative stress actually affects the mitochondria itself. So some mitochondria go bad and some are good in the cell. So these bad mitochondria need to be filtered from the cell, removed from the cell, because they have a potential to cause apoptosis. So mitochondria can directly activate apoptosis, and this is the reason why the damaged mitochondria need to be cleared from the cellular system. So PINK1 is a quality controller. So I'll briefly explain you the mechanism. I'm sorry, this is a complicated slide over here, but this is the mechanism how PINK1 functions. So this is the PINK1 protein, and it binds to the outer mitochondrial membrane of the mitochondria. So when the mitochondria is in a healthy state, so as you know, mitochondria synthesizes ATP, and this also generates a potential, a membrane potential, like a gradient. So this membrane potential is very important for import of proteins and various other functions of mitochondria. And when mitochondria is suffering from some problems, this mitochondrial membrane potential goes bad. So the mitochondrial membrane got depolarized. So PINK1 is actually kind of a quality controller here. It checks the membrane potential of mitochondria here. So it binds to the outer mitochondrial membrane, and it senses whether the proteins are synthesized in cytosol, and it is imported into mitochondria. As you know, majority of the proteins which are required for mitochondrial function are actually imported into mitochondria. Mitochondria has its own genome, but it only codes for few of the mitochondrial proteins. So PINK1 senses this change in protein input, and it aggregates in the outer mitochondrial membranes. The aggregation causes phosphorylation of other proteins such as parkin and ubiquitin, and the phosphorylation by PINK1 on parkin and ubiquitin initiates the process of mitophagy. So mitophagy is initiated, and the next stage is assembly of phagocytosomes. So the autophagy machinery kicks in. So as you know, the lysosomes comes and it engulfs the damaged mitochondria, and it rescues the cell from the bad effects of this mitochondria. So PINK1 is kind of a quality controller here. It checks whether the mitochondria is good or bad. So just to recap what you have learned till now. So Parkinson's disease is a neurodegenerative disease. Dopamine producing neurons are lost in Parkinson's disease, and the lack of dopamine causes motor as well as non-motor symptoms in PD patients. And there is no cure for PD, and the present treatment is symptomatic. So apart from that, the genetic mutations which causes almost 20% of PD, but these studying these genes which are loci for mutation and which are responsible for causing the disease, is an essential tool to study Parkinson's disease and to know the molecular mechanism, especially because the various molecular mechanisms of the disease, the pathological mechanisms, are very similar between sporadic and familial Parkinson's disease. So mitochondrial dysfunction is a common similarity, as I told you before, and it's often associated with different kinds of PD pathology. And loss of function mutation in PINK1, which is my focus, causes Parkinson's disease in humans. So I'm going to split my talk here into two sections here. So one of the talk will be concentrating on mitochondrial calcium homeostasis, which is disturbed in PINK1 deficient condition. And the second topic would be extra mitochondrial roles of PINK1 in neuronal development. So these were two different projects which I have done during my stay in Europe, and the next one was in the USA. So the first project, mitochondrial calcium homeostasis, is disturbed in PINK1 deficient zebrafish. What you see here is a PINK1 zebrafish larvae, which is 5-day-old zebrafish larvae, and it is the neurons are tagged with GFP. So the whole green, the bright greens, are actually neurons. So you can see this is the brain and this is the spinal cord. Zebrafish is an excellent model to study neurodegenerative diseases. One of the main reasons is because the zebrafish's embryos or larvae are transparent. So you can check the expression levels of your desired proteins through microscopy, and you can get in-depth images and in-depth expression analysis. So this is these are zebrafish tanks. So one of the bigger zebrafish tanks can hold at least 100 zebrafish, and if you make these zebrafish, you can get like around 400 embryos, and you can work with those embryos. So it is much more cheaper than working with a mice model or a rat model. And as I told you, the fertilization is external. So you pair your fish, you have embryos. So it's an external fertilization is less complicated. And another main important factor is that 70% of the human genes are conserved in zebrafish, and around 80% of human disease genes are as well conserved. So you can study a lot the mechanism of disease because the genes are conserved, their function is conserved. So why calcium signaling? Why is calcium signaling important? So calcium is essential for neuronal function. You cannot work without calcium. It is a secondary messenger which is involved in everything from fertilization, neuronal development, survival, everything. Everywhere calcium is there. So in the context of neurons, it is important for membrane excitability, neuronal transmission. Many of the proteins are activated by calcium, such as proteases, certain kinases, certain phosphatases. Gene expression is regulated by calcium. Mitochondrial function is another important role of calcium because for the proper functioning of ATP synthase, which synthesizes ATP, which is the fourth complex in the mitochondria, you need calcium for that activity. Unfortunately, there is also a bad effect of calcium. There should be a proper regulation of calcium levels inside the cell. So the cell has many stores for calcium. The biggest store is endoplasmic reticulum, and various cell signaling mechanisms control the release of calcium from these stores like endoplasmic reticulum and mitochondria, and govern the whole cellular function. So as I told you, there are two main stores, endoplasmic reticulum and mitochondria. You may need to remember that for the next slide. And another major important role of calcium is apoptosis. Apoptosis is also involved regulation of calcium signaling. So there is a huge mechanism inside a cell to regulate calcium levels so that various functions can be switched on or switched off, but there should be a balance. And the balance is kind of lost in certain cases of Parkinson's disease. I'm sorry about this slide. This was supposed to work with the animation. So dopaminergic neurons are kind of unique. Dopaminergic neurons, which I had told you, which are in the substantia nigra, they're kind of unique because they employ calcium-dependent channels. So they're called LTCC. So these channels are on the outer cellular membrane, and they bring in calcium into the cell. Usually in neurons, the neuronal activity, the neuronal transmission, is mainly regulated by sodium and potassium ions. But dopaminergic neurons, to a certain extent, calcium also regulates neuron. Calcium is also responsible, not regulates, it's responsible for maintaining that pace-keeping activity which is required for neuronal transmission. So what it means that unlike other neurons, the neurons in the dopaminergic neurons have a high level of calcium. And as I told you, high levels of calcium must be regulated, otherwise it could be fatal to the cell. So apart from that, dopaminergic neurons are very extensive neurons. They have huge dendrites. So there's a huge energy demand. That means that the mitochondria is functioning on its maximum output. And that also increases free radicals in the mitochondria. So there is a connection between mitochondrial calcium overload and the pathology which is seen in Parkinson's disease. And I'm interested to study that. So this is a brief explanation of how the mitochondrial calcium setup works. So there are there are channels on the outer mitochondrial membrane. This is called VDAC, which brings in calcium out from the cell cytosol, from the cytoplasm into the mitochondria. So, and then there is mitochondrial calcium uniporter, which is found in the inner mitochondrial membrane, and it's very selective in nature, and it mainly is responsible for transporting high calcium concentration. It works during high calcium concentration. So my hypothesis was that during PINK1 deficiency, or you have, for example, a PD patient, and there is a PINK1 mutations in them. In their neurons, there is a mitochondrial calcium overload. And how can we stop this overload? How can we regulate this mitochondrial calcium overload? So what we can do is we can inhibit the function of mitochondrial calcium uniporter, which brings in higher concentration of calcium into the mitochondria, and and changes the balance in calcium in the mitochondria. And so that that change in the balance is restored if we can alter the mitochondrial calcium uniporter. So the hypothesis was that. So I started working with PINK1 mutant zebrafish. So in this particular zebrafish, this was developed in University of Sheffield. This particular zebrafish model, which has a mutation in its PINK1 gene. So the PINK1 gene is not functional. So the protein is not functional. And as you can see, due to the loss of PINK1, there is a decrease in dopaminergic neurons. So the green ones, the green labeled cell neurons, are the dopaminergic neurons. And this is the zebrafish larvae. The brain of the zebrafish larvae, which has been dissected from the embryo, and then they have been treated with antibodies specific for dopaminergic cells, which are tyrosine hydroxylase. And as you can see, that there is a decrease in dopaminergic neurons in the PINK1 mutant zebrafish when compared to the wild type. And this is one of the major reasons in humans what we see, that there's a loss of dopaminergic neurons, and we have replicated that in the zebrafish model. So the next step was to check whether there is any imbalance in mitochondrial calcium. We had to check the mitochondria, whether there is a mitochondrial calcium overload. So this is a 5-day-old zebrafish embryo, and it has been tagged with a GCAM. So GCAM is a genetically encoded calcium indicator. So it senses the change in calcium concentration. And in this particular model, what we saw is that there is an increase in mitochondrial calcium levels, which you can see here in the PINK1 mutant zebrafish. So this indicates that PINK1 mutant zebrafish have an increase in mitochondrial calcium in their mitochondria, which functions as a mitochondrial calcium overload and has negative effects because of that. So the next step was, how would we inhibit the effect of mitochondrial calcium uniporter? So preliminarily, we did morpholino studies. So morpholinos are kind of oligonucleotides, and they bind to the mRNA, which is going to be spliced, and it inhibits the splicing. You must have studied mRNA splicing. It's part of the maturation of the RNA, right? So it blocks the splicing. So when the splicing is blocked, the mRNA is not developed. It's not processed in a proper manner. So this is a control. So here there is no morpholino treated, and in the morpholino treated zebrafish, you can see that there are multiple bands. What this indicates that the mRNA has splicing has not been done properly. So there are different kinds of mRNA, and because of this, the mRNA go into something called as an mRNA decay. So what happens is that the mRNA which have improper splicing, improper exons in their framework, they degrade because the cell senses that mRNA is not a physiological mRNA, and thereby it degrades the mRNA. So this is a quantitative PCR, and you can see that there's a lower levels of the mitochondrial calcium unipotent. So what is the effect of morpholino? So I just want to recap. We did the inhibition of mitochondrial calcium unipotent, and we wanted to check what will be the effect on dopaminergic neurons. So your mitochondrial calcium unipotent is inhibited, mitochondrial calcium influx, so the calcium coming into the mitochondria is decreased, and what would be the effect of the decreased calcium levels in mitochondria? So what we saw is that, so these are wild type zebrafish embryos, and these are the dopaminergic neurons stains, the mRNA stains. So this in situ hybridization, and as you can see in the PINK1 condition, there's a decreased number of dopaminergic cells, dopaminergic neurons. And what happens is that in the morpholino injected zebrafish, there is a rescue of the dopaminergic neurons. So PINK1 mutant zebrafish, which were injected with MCU morpholino, which blocks MCU function, that causes the rescue of dopaminergic neurons. So this indicates that mitochondrial calcium overload is not only present in PINK1 mutant zebrafish, but if you inhibit it by using morpholinos, you can actually get a rescue effect in the neurons. So the neurons get better. So this nudged us to use CRISPR-Cas9 to generate a MCU knockout. So the difference between a knockout and a knockdown is in the knockdown, where I used morpholinos, the levels of mRNA decrease, right? When the levels of mRNA decrease, of course, the protein also decreases. But when we use CRISPR-Cas9 and we create a stable mutant, what happens is that the MCU levels is completely deplenished from the embryo. So these are stable knockouts. So the protein is almost also inhibited because there's an indel mutation. So insertion of a oligonucleotide and deletion of oligonucleotide from the cDNA of the MCU gene, and this causes a truncated version, a smaller version of the gene, and this results in altered protein. So here you can see this is the agarose gel electrophoresis of the MCU knockout zebrafish genome. So this is you take the genomic DNA and you do the PCR, and this is from the different populations of your CRISPR-Cas9 treated zebrafish. So the wild type, this is the wild type allele. There is no mutation, and it is in the proper place. But this is the MCU heterozygous. So there is a wild type allele as well as a mutant allele, which you can see here. And this is the MCU homozygous zebrafish, which only has a mutant allele. And what happens is that because this MCU is a mutant allele, there is a indel mutation. There is an RNA decay also happening. So the MCU protein remains non-functional. We wanted to check whether the MCU protein is non-functional, and what we did is that we used some fluorescent calcium reporters inside the cell, and we checked whether the calcium influx is blocked in the mitochondrial calcium unipotent knockout zebrafish. So as you can see, the green line here, it is the line from the wild type zebrafish where there is a influx of mitochondrial calcium, but in the MCU zebrafish, there is no influx happening, and this is confirmed by a pharmacological inhibitor of mitochondrial calcium reporter, which is called ruthenium red. So ruthenium red, when we treat the cells with ruthenium red, mitochondrial calcium reporter is blocked, and as you can see, the knockout and the pharmacological inhibitor had the same effect. So we were quite happy with that, and it means that the model is functioning and there is no influx happening. So we checked and we used immunohistochemistry to check the dopaminergic neurons in the zebrafish. So here you can see these are dopaminergic neurons labeled by thyroxine hydroxylase antibody, and as you can see, there is a decrease in dopaminergic neurons in the PINK1 mutant zebrafish, and when you have a knockout of mitochondrial calcium uniporter in the PINK1 condition, there is a rescue of the dopaminergic neurons happening. This is the analysis using a software called as Imaris. So we know that the number of dopaminergic neurons are decreased in PINK1 knockout zebrafish, but when the MCU is inactivated, there is a rescue happening of dopaminergic neurons. These are new dopaminergic neurons which also have been labeled by a mitochondrial specific dye, and as you can see that there is a decreased volume of the mitochondria in the PINK1 mutant zebrafish. What happens is that the mitochondria are kind of fused and they are not in a healthy state. But what happens in the PINK1 MCU double knockout zebrafish is that the phenotype is rescued and the mitochondria is in a functional state. So part one, conclusion. So what we learned from the first project. So we learned that PINK1 deficient zebrafish are prone to toxicity due to mitochondrial calcium overload, and this mitochondrial calcium overload is present in PINK1 mutant zebrafish, and mitochondrial calcium influx can be stopped by MCU knockout. And if you knock out the MCU in the PINK1 mutant zebrafish, you get a rescue of the dopaminergic neurons in the zebrafish model of Parkinson's disease. So these are primary cortical neurons. So we extract cortical neurons from mouse embryos and then we plate them on on culture dishes, and then we grow them to do our experiments. So here you can see neurons which are labeled by MAP2B, which is a pan-neurite marker, and you can see the nucleus. The red one is actually a synaptic marker, synaptophysin. So the second project dealt with, excuse me. So the second project dealt with extramitochondrial effects of PINK1 in neuronal development. So this is the city where I live. This is Reno, Nevada, and there's a beautiful lake around 40 miles from here. It's called Lake Tahoe. I think it's one of the largest alpine lakes in the U.S. And this is my research group with my supervisor and my other colleagues, and this is our university. Just to give a break in between all the data slides. All right, going back to data. So I just wanted to recap. So you all should be aware of PINK1 right now. You have a brief understanding of PINK1. It's a kinase. It phosphorylates certain proteins which have functions in various mitochondrial functions. And apart from that, there are also some extra mitochondrial functions. So PINK1 phosphorylates Parkin, which I told you before, and it just has a role in mitophagy. Ubiquitin, BCL2. BCL2 is another protein which is involved in anti-apoptosis. So PINK1 becomes neuroprotective by its action. It also activates protein kinase A, which is a master regulator. It's part of the cyclic AMP PK signaling pathway, and it also phosphorylates MERO, which is involved in mitochondrial transport. So mitochondria are dynamic organelles which move around the neurons. It goes into dendrites, goes into axon. Wherever energy is required, wherever calcium sequestration is required, mitochondria has to be there. So extensive cellular machinery is involved in transporting mitochondria across different domains. So as I told you, PINK1 is mainly involved in clearing defective mitochondria, the process called as mitophagy. And that is the canonical role. That's the conventional role of PINK1. But the lab which I'm currently working on, we are interested in looking the non-canonical role of PINK1 in mitophagy. So there are certain roles attributed to PINK1 which are not related to mitophagy. We are interested in that. So why there exists a non-canonical role? Because uh, this PINK1 protein kind of has a diabolical role. It functions in a different way. So, for example, this is a full-length PINK1, and there's a conditional proteolytic processing happening. So, as I told you before, during loss of mitochondrial membrane potential, mitochondria goes bad, the defective mitochondria, the PINK1 comes and aggregates. But what happens in a healthy state, right? Because this is a dynamic process. This is happening all throughout the time. PINK1 is coming, it is binding to the outer mitochondrial membrane, it is checking out whether the mitochondria is doing well or not. And if it is not doing well, it starts the mitophagy. But what happens otherwise? So that's what we're interested in. So what happens is that full-length PINK1 checks the membrane potential, checks the protein transport. If the protein transport is fine, the there's a sequence in the PINK1 protein. It's called the mitochondrial. It's a mitochondrial signal. So what happens is that this particular sequence of protein, this sequence enables the proteins to be targeted to the mitochondria. So this particular sequence, the mitochondrial localization sequence, this is cleaved on the outer mitochondrial membrane, and the cleaved PINK1 translocates to the cytoplasm. So usually, full-length PINK1, canonically speaking, is associated with mitochondria. But after this cleavage, cleaved PINK1 localizes in cytoplasm. So what that indicates that cleaved PINK1 could have some role in the cytoplasm in regulating cellular signaling. So we are mostly interested to figure out what are those particular pathways which cleaved PINK1 affects. So there was a photo here which I cannot show you right now because of the animation thing. So what happens is a full-length PINK1 is around 68 kilodalton long PINK1, a long protein. And cleaved PINK1, there are multiple cleaved PINK1s. One is a 54, 52, one is a 48 kilodalton. So there are different sizes of proteins. And these cleaved PINK1 are kind of seen in the cytoplasm. So one of the major findings that we saw is that during neuronal development, the full-length PINK1 remains stable. We cannot see a change in expression levels of full-length PINK1. But what happens in cleaved PINK1 is that there is an aggregation of cleaved PINK1s throughout neuronal development, which indicates that cleaved PINK1 could have certain roles in neuronal development. So we did a Western blotting of cleaved PINK1 from the protein isolated from different stages of neurons. And what we saw is that cleaved PINK1 aggregates during neuronal development, and which is similar to increased expression levels of proteins such as BDNF, which is required, which is essential for neuronal development. In this particular side, what you see is like these are SHS5 cells, these are neuroblastoma cells, and neuroblastoma cells, when they are treated with retinoic acid, they turn into dopaminergic neurons. So this is one of the a model which is heavily used in neurobiology research. So what we saw is that when we treat the SHSY cells with kinetin, so kinetin is a pharmacological activator of PINK1. So it's a plant hormone basically, but it is an analog of ATP. So the structure is similar to ATP, and it lends a phosphate group to PINK1, activating the PINK1. So it's an activator of PINK1. So what we saw is that just like retinoic acid, when you give kinetin, there is an increased expression level of cleaved PINK1. So both retinoic acid and kinetin causes increased expression level of cleaved PINK1, which indicates that cleaved PINK1 could have some major role in neuronal development or differentiation. So this was one of the starting points. So as I had told you before, we checked also the level. We checked on the levels of certain neurotrophic factor called as BDNF, and it matched with the cleaved PINK1. So we were wondering whether what happens with BDNF, what happens about BDNF levels in PINK1 deficient condition. So these are midbrain sections from wild type, the control, and the PINK1 knockout mouse. What happens is that there is a decrease in BDNF levels in the dopaminergic neurons. So the red ones are the dopaminergic neurons, and there is a decrease in BDNF levels in the dopaminergic neurons. We also check this in cortical neurons, primary cortical neurons, and we see a decrease in BDNF expression over there as well. So the immediately we concluded that there is a link. There is a link between PINK1 and BDNF function. So BDNF, what is BDNF? BDNF or brain-derived neurotrophic factor. It is an essential neurotrophic factor, and it is required for neuronal growth, differentiation, maturation, maintenance, and survival. When you grow neurons on a dish, you need to add BDNF as a supplement for the proper growth of the neurons. So it is as essential as that. This is a Western blot of various lysates from primary cortical neurons. So the primary cortical neurons are grown on a plate. You change the media and you let it grow for a point of time when the neurons are matured. You lyse the neurons and extract proteins from them, and you run Western blotting. Western blotting is a technique to estimate the protein levels. So here, what you can see is that the the cells which were treated with kinetin for 20 24 hours, there is an increased expression level of mature BDNF. So what this indicates that when you treat your cells with kinetin, which activates PINK1, it leads to an increased level of BDNF. So there is a direct relation between PINK1 activation and BDNF levels. All right. So in the next step, we wanted to know whether cleaved PINK1 has any particular role. So we did multiple transfections. So here we transfect primary cortical neurons, PINK1 primary cortical, knockout primary cortical. So what happens here is that these particular primary cortical neurons, they don't have a functional PINK1 gene, right? So there is no PINK1 protein. So PINK1 is absent in these cells. So then we reintroduce PINK1 through plasmids. For example, here we reintroduce full-length PINK1, and here we introduce cleaved PINK1. So the delta and sign is for a cleaved PINK1. What happens is that when we transfect these cells with full-length PINK1, we don't see an increase in BDNF levels. But when we transfect these cells with the cleaved form of PINK1, we see increased level of BDNF. So this is a direct evidence which shows that cleaved PINK1 is involved in the enhanced level of BDNF. At the time when we were doing the experiment, we also saw that apart from the neurons which are transfected with cleaved PINK1, the neurons around that cleaved PINK1 are around that transfected I would rephrase that. So here, as you can see, there are two transfections happening. There's a control and there's a delta PINK1. What happens is that when a cleaved PINK1 is transfected, the neurons adjacent to the transfected neurons, they are also showing increased BDNF levels. So we thought that apart from enhancing BDNF levels, maybe there is some enhanced BDNF secretion also happening. So here we did an ELISA assay and we checked the BDNF levels in the media. So we took the media and filtered it and then checked the levels of BDNF through a ELISA assay. And we saw that just like cyclic AMP, so cyclic AMP is involved in the PK pathway, and it directly increases the levels of BDNF. So this is kind of a positive control. So as a positive control, it means that you will definitely see the result here. So you should definitely see what you want to see here. And you compare it with your treatment, and as you can see, when you treat with kinetin, there is an increase in BDNF levels over there, which we saw. So now we know that PINK1 affects BDNF function. We need to know the mechanism. How does PINK1 affect PK function? BDNF function. So BDNF is a protein which is heavily involved in neurotrophic signaling, as I told you. And it is also governed, the levels of BDNF is also governed by a pathway called a cyclic AMP PK pathway. Cyclic AMP is, as you know, it's a GPCR thing, and it's a second messenger. It activates protein kinase A, and protein kinase A goes into the nucleus and it activates CREB. CREB is translocated. CREB is a transcription factor which increases the levels of BDNF. And we were checking the levels and activation of all these particular parameters throughout neuronal development. And what we saw is that PK is also getting activated by kinetin. As you can see, this is a phosphorylated PKA. This is a Western blot where we use phosphorylated antibody. So this antibody binds to the phosphospecific region, which means that only if the PK is phosphorylated, this will show you a result. And as you can see, there's an increased levels of the phospho-PKA here, which indicates that your treatment is this phosphorylation. So what we see here is that PINK1 activation causes phosphorylation of protein kinase A. And this in turn results in the subsequent cellular signaling. As I told you before, the PINK1 activation causes PK activation. The subsequent effect of PK activation is CREB activation, and we wanted to check that. And as you can see, there's an increase in CREB localization in the nucleus, and this indicates that CREB is entering into the nucleus and increasing the transcription of the BDNF. So here, there is a Western blot assay of the similar. This is the immunofluorescence assay, and in both you can see similar results. So what is the end effect? What is the end effect of all of this, right? PINK1 activates PK, PK activates CREB. What happens next? So what happens is that there is an increase in neuronal plasticity. And how did we conclude that? So we checked the expression level of various markers of these neuronal plasticity, such as PSD95, synaptophysin. So these are proteins which have functions in neuronal plasticity. But not only that, they are localized. They are localized on the spines, so they are localized near the synapses, right? So, for example, this is a dendrite, and as you can see, these are various spines, and these are the synapses over here. You can see the bulb-like structures. These are the synapses. So, in our results, what we saw is that when we treat the cells with kinetin, which activates PINK1, which activates the PKA CREB signaling, it results in increased expression of PSD level. There is increased synapses. As you can see, these are the synapses which has been quantified on the dendrites, and there is an increased synapses, increased expression of PSD95 on the dendrites. Similarly, these are synaptophysin which are on the axons. So you can see that there's an increased level of the synaptic proteins, which indicates that there's an increase in neuronal plasticity. So what happens after that? So the increased synaptic plasticity means there are more synapses. The neuron wants to make more contacts, and the dendrite arborization, the whole network of dendrites increases. So here, as you can see, treatment with 24 hours of kinetin, which activates PINK1, causes increased arborization of dendrites. The neurites are much more complicated. They're elongated. The density is increased. So this is one of the end results of activation of PINK1. This is a complicated slide. I'm sorry, and I'll not explain everything over here. I'll just go through the main things here. So full-length PINK1, as I told you, there's a conditional proteolytic processing. There's a retrotranslocation. It translocates to cytoplasm. There it is stabilized, where certain chaperones are involved in stabilization of this cleaved form of PINK1. Cleaved PINK1 then exerts its influence. The one thing I think I have not mentioned is that the cleaved PINK1 still retains the kinase domain. So it still can phosphorylate other proteins. So here it phosphorylates PK. PK activation leads to CREB activation. CREB goes into nucleus, and there is an increase in expression of BDNF likewise. What is the effect of all of this? So, as I told you, there is an increased neuronal plasticity through the increased BDNF regulation, and as you can see, that's over here. Apart from this particular pathway which I had described you right now, which we found, there are also other pathways in which cytoplasmic PINK1 function, including mitochondrial transport, anti-apoptosis, and degradation as well of some certain other proteins, which I'm not going into right now because of lack of time. So what we learned from the second plot. So BDNF function is impaired in PINK1 deficient condition, and stimulating PINK1 function causes BDNF levels and secretion to increase. Cleaved PINK1 but not full-length PINK1 stimulated BDNF function, and PINK1 exerts its influence on BDNF through activating PKA-CREB signaling. So this novel pathway is an example of mitochondrial retrograde pathway. By retrograde pathway, it means that mitochondria controls cellular signaling through its own state, because the mitochondrial it is dependent on the mitochondrial membrane potential. So the state of the mitochondria signals other pathways inside the cell. If you want to learn more about these studies, you can check these research articles. You can also go to Google Scholar and you can type in my name and you can find other papers which I have published, and you can learn more about that. So I want to acknowledge now. So I want to acknowledge Dr. Ruben Daka. He is my current supervisor, and I want to acknowledge other people also involved during my previous career stages. And of course, I want to acknowledge the funding agencies, National Institute of Health. We are working on R1 project, National Science Center Poland. I was a principal investigator in one of the grants over there. Parkinson's UK. My previous supervisor, Dr. Banman, got grants from there, which we were working on. And of course, University of Reno, which was supporting all the support. All right, so the science part of the talk is over. I'm just going to give you some brief inputs into career in science. So as I understand, most of you who are listening to this talk, you are MSc students, and after an MSc, if you are interested into research, you can go into doctoral studies. You can take a PhD, and then you can go through the ladder of academia or industry as you want. There are different careers after a PhD. If you want to stay in academics after a PhD, you do a postdoctoral fellowship. You get more training. You get specialized in certain techniques, not only techniques, in research teams, and then subsequently you go into an assistant professorship, associate professorship. You have to write grants. You have to be very patient. You have to be passionate about science, because staying in academia doing research is not always fun, because most of the time you're working with negative results. You don't get anything. Nothing works. But when it works, enjoy that and go through it. Go through the grind. So staying in research needs a lot of passion. So if you want to get into that, you need to know that. Apart from academia, there's also option for industry. There are a variety of biotechnological and pharmaceutical companies which recruit scientists who have PhD, also MSc, and you can work in research projects over there, developing therapeutics for neurodegeneration, cancer biology, etc. Apart from that, you can get into science policy, develop how a policy of the nation regarding science works. There are a lot of agencies doing that. You can get into that. You can get into scientific writing. There are a lot of opportunities over there as well. And of course, there's intellectual property as well. You can be involved in that as well. So there are multiple opportunities for students who have completed their masters, who want to do research and continue their career in a PhD and after getting a PhD. So PhD, of course, in India or abroad, you need to have a MSc or a BTech. With the BTech, you can get PhD in Europe and USA as well. But in India, as I understand, you need an MSc. But I'm not sure. Okay, you have to check that. So one of the most important thing is find a topic which really interests you, which you are curious about, and you want to work on it. If you want to work on it, you have to be sincere on it, and it means a lot for you, that topic. Another is find a good mentor. This is one of the most important things. Even if uh, the topic matches you and the mentor is not good enough, that's a bad choice. So you need to find a good mentor whom you can trust and who you believe will help you in your career development, in making you a good research scientist. Apart from that, you have to try for various research fellowships after MSc, such as CSIR, DBT, and other fellowships. And there are many recent ones as well, such as the Prime Minister Research Fellowship and stuff like that. So after this fellowship, if you want to do your PhD back in India, you can get into premier institutes. There are a lot of good institutes in India doing very good science, such as Indian Institute of Science, NCBS, and a lot of others. These are just examples. So get in there. If you want to do your PhD there, do your PhD there, and move in a direction for a postdoctoral fellowship or a career in industry or academia. If you want to do a PhD abroad, in the case of USA, you have to clear the GRE. That's the first step. Then you have to find positions in the universities which are advertised in various portals such as Nature Jobs, Science Jobs. You can check into university websites as well, and you can apply there. Once you apply, and if you are selected, you need to take a lot of courses. Unlike in India, you don't have that much of a coursework doing a PhD, but in the USA especially, there's a lot of coursework involved. So you have to devote some time, especially during the first few years, for taking courses, taking exams as well, apart from doing science. PhD in USA is around 4 to 6 years. It takes time. After you finish your PhD, as I told you, there are multiple opportunities in academia, industry, government jobs, policy making, etc. I wish you all the best if you want to stay in industry, if you want to stay in academia, if you want to stay in research. It's a very good opportunity. The life is good. There are a lot of perks. So that's it. That's it from me now. I would like to thank all of you for your attention. So this is I'll part away with a zebrafish selfie. This is an award. It's an award-winning photo taken by Dr. Oscar Ruiz of a zebrafish larvae. These are not the eyes. These are the nostrils. But this is the face. All right. Thank you very much. I'm happy to take questions, and you can contact me for any further information if you want. Thank you.
Thank you, Smritin. It's so nice to have you here.
So nice to see you, Sally, ma'am.
So it's my privilege to have been asked to propose a vote of thanks on this occasion. I, on behalf of my department, Biotechnology Department, extend a very heartfelt thanks to Dr. Smritin Suman, who honored us with his presence and took out his valuable time of his busy schedule. Thank you, Dr. Suman. I would like to especially thank your efforts which you took in preparing for this presentation, for it enlightening us with this complexity of Parkinson's disease in so precise manner. I also wish to express my thanks and gratitude to our principal ma'am, Dr. Shaljapan, for providing encouragement and support. I am very grateful to Chairman Sir Shri Arvind Gupta ji for giving us an opportunity to organize this event. I heartily congratulate to all the participants for their active participation in this event. And this program would not be successful without my fellow colleagues, Ms. Dipti Gulati and Dr. Manisha Nanda, and our technical team for their support. Thank you all for making this event successful and for your contribution also. Thank you, everyone. Thank you, Smritin. Thank you so much for sparing your time.
Thank you, Sally, ma'am. It was fun. It was a nice opportunity. It was nice
So
to
I
see
hope
you
you
all.
revive your memories.
Yes, I didn't see the students, But yeah, after seeing you and Deepti ma'am, yeah,
Yes, there was
obviously.
some technical issues, so we could not switch on the video here.
Right, right. No
We
problem,
would
no
like
problem.
to have you again here for an insightful talk.
Yeah, sure. Thank you, thank you,
Thank you so much.
thank you.
And